Yes, my dear student, now we will discuss the new topic that is known as the transcription. The process of copying genetic information from one strand of the DNA into RNA is termed as the transcription. It means the DNA, their information is simply transformed in the form of messenger RNA, which helps into the formation of protein synthesis or polypeptide chain. So we can say that the process of copying genetic information from one strand of the DNA into RNA is termed as the transcription. The principle of complementary governs the process, except that adenosine now base pairs with uracil instead of thymine as in replication. As you have learned that the thymine is replaced by the uracil in case of the RNA. Unlike replication, only a single stranded fragment of DNA gets copied into DNA. As you know that DNA is double stranded, but at the time of the transcription, when there is the information is transcribed, it will be done by only one means strand of the DNA. If both now the question is that what happened if both the strands will code for RNA? Two different means complementary RNA molecules and two different proteins would be formed. Hence, the genetic information transfer machinery would become complicated. So, since the two RNA molecules produced would be complementary to each other, they would bind together to form a double-stranded RNA without carrying the translation. That means the process of transcription would become futile. It is not possible. So only one strand of the DNA take part into the transcription process. Yes, my dear student, just you see the transcription unit. The transcription unit of DNA contains three regions in the DNA: the promoter, a structural gene, and a terminator. Let us understand. It is the promoter region. Here it is the structural gene and it is the terminator. That place is known as the terminator. What is the role of one by one? Let us understand. The promoter, it is the binding site for RNA polymerase for initiation of transcription. Means when there is the transcription, this promoter allow the RNA polymerase to bind. The structural gene, it codes for enzyme or proteins for structural functions. The terminator gene, what is its role? It is the region where the transcription ends. The DNA-dependent RNA polymerase helps in DNA replication by catalyzing the polymerization in only one direction, that is 5' to 3' direction. As you have learned just now that the DNA replication is also followed in one strand which have polarity 5' to 3' direction. The DNA strand that has the polarity 3' to 5' direction act as a template and is also referred as the template strand. The strand which does not get transcribed is called the coding strand and has the polarity 5' to 3'. Its sequence is same as an RNA. The promoter gene is located towards the 5' end, that is known as the upstream of the structural gene of coding strands and provide the binding site for RNA polymerase. The sequence of DNA located towards the 3' end downstream of the coding strand where the process of transcription would stop. It is known as the terminator. Now, the transcription unit and the gene. Let us understand what is the transcription unit and the gene. The segment of DNA coding for a polypeptide is called cistron. In eukaryotes, The transcription unit possesses a structural gene, a specific only for a single polypeptide chain. Thus, it is called monocistronic. In prokaryotes, the transcription unit possessing the structural gene, it is known as the polycistronic. The gene in eukaryotes are split into the coding or express sequence of DNA, which is known as the exon. The gene in eukaryotes are split into coding or express sequence of DNA called the exon, and non-expressible sequence of DNA is known as the introns. So my dear students, here you have to see, you have to learn the 2 points. One is known as the exon, means coding sequence. Non-coding sequence will be known as the introns. Messenger RNA contains only exon but no introns. Now when we will see in the process how there is a transcription takes place in eukaryotes and what is the role of this exon, how the introns will be removed, so we will discuss into the next